Hello students, today we will be studying epithelium part 1. Now let's first see the structural organization of the body. So the entire human body has a number of organ systems like respiratory system, cardiovascular system, each system having a number of organs and each organ having different tissues and each tissue made up of different types of cells. Now let us first see introduction of epithelium. How do we define a tissue? Cells and extracellular components having similar morphological characters and organized in different proportions and patterns to perform specific function is called as a tissue. Or in a simple way we can say collection of cells of similar structure to perform a specific function is called as tissue. And as we know, there are 4 basic tissues in the body: epithelial tissue, connective tissue, muscular tissue and nervous tissue. So epithelium is one of the basic tissues of the body. Singular epithelium, plural is epithelia. Epithelium covers the outer surface of the body and also lines the internal surface of all the body cavities. Now, how is the word epithelium derived? Epi meaning upon and thele meaning nipple or papilla. Hence, the lining on the papillae of the lips was first called epithelium by Ruisch in 1700. Now, how do we define epithelium? So, epithelial tissues are composed of closely aggregated cells with very little extracellular substance, and they cover internal or external surface. Now these cells have strong adhesion due to adhesion molecules, membrane interdigitations and intercellular junctions. Now let us study the forms and characteristics of the epithelial cells. Epithelial cells are adherent to each other by means of junctional complexes. Very little intracellular material is found between the epithelial cells. The deep surface of epithelium rests on basement membrane which separates the epithelium from the underlying connective tissue. The superficial surface of the epithelium is exposed to air or fluid and often shows modifications. Epithelium does not have blood vessels and lymphatics. Nourishment to the epithelium is provided by diffusion from adjacent supporting tissue. And epithelium has a very good regenerative power. The shape of the nucleus of the epithelial cells corresponds to the shape of the cell of the epithelium. Thus, columnar cells have an oval nucleus, cuboidal cells have spherical nuclei, and squamous cells have flattened nuclei. And the long axis of the nucleus is always parallel to the main axis of the cell. Now, as we see here, a diagram of epithelium, this is the basement membrane. These are epithelial cells of the basal layer. As we can see that the nucleus is oval, so we assume that the cells are going to be columnar. Similarly here the cells are showing a rounded nucleus, so these cells will be polyhedral or cuboidal cells. Epithelium invaginates and grows in the underlying tissue and is specialized as glands. Epithelium is derived from all the 3 germ layers. The ectoderm giving rise to the epithelium of the skin, that is the epidermis. Endoderm gives rise to the lining epithelium of the respiratory and the digestive systems, and mesoderm gives rise to the cardiovascular system. The forms and dimensions of epithelial cells range from high columnar to cuboidal to low squamous cells. Because the boundaries between cells are frequently indistinguishable with the light microscope, the form of the cell nucleus is an important clue to the shape and number of cells. Nuclear form is also of value in determining whether the cells are arranged in layers, a primary morphological criteria for classifying epithelium. So if we see 2 or 3 rows of nuclei, we can assume that it is a multi-layered epithelium. Now let's study the basement membrane. Almost all epithelial cells, whether lining a surface or forming gland units, rest on a basement membrane which is made up of connective tissue. The portion of the epithelial cell that faces the basement membrane is called the basal pore. So what we see here, this is the basement membrane, and these are the cells which rest on the basement membrane. So this surface, that is the surface which faces the basement membrane, is called the basal pore. The opposite side, usually facing a space, is called the apical pore. So this is the opposite surface. This would be the apical pore of these cells. And the surface of the apical pore is also called as the free surface because it is facing the exterior, or it is facing the lumen of a tubular structure. Whereas surfaces that are opposed to neighboring cells are called as lateral surfaces. So these surfaces here would be the lateral surfaces. So basal surface, apical or free surface, and these are the lateral surfaces. Now we go on to study some surface modifications of epithelium. Epithelial cells have some surface modifications, and these are divided into apical surface modifications and lateral surface modifications. Let us study the apical surface modifications. The apical surface of the epithelium is facing the lumen, and apical surface modifications are necessary for epithelium to carry out various functions. So the apical modifications are of 4 types: microvilli, stereocilia, cilia, and flagella. Microvilli, singular, being microvillus. Microvilli, Greek word micros meaning small and villus meaning tuft of hair. Microvilli are cylindrical, membrane-bound projections of the cytoplasm from the apical or luminal surface of the cells. They represent a striated border of the intestinal absorptive cells and brush border of the kidney proximal tubule cells, which are observed under the light microscope. What we see here is a columnar cell having microvilli, projections seen on the apical surface. If these microvilli are arranged in a regular fashion, they form what is known as a striated border. And if they are irregularly situated on the apical surface, then they give rise to the brush border appearance. The complex of microvilli and glycocalyx is called the brush or the striated border because in these absorptive cells, the glycocalyx is thicker than it is in most other cells. The length of microvilli is between 0.5 to 2 micrometers. Central core of microvilli contains the microfilaments. This here is a transverse section passing through a microvillus. So the central core here is made up of microfilaments, and what we see here is the outer cell membrane. Microvilli increase the surface area of absorption. So function of microvilli is absorption. And where do we see microvilli? Intestinal epithelium and proximal convoluted tubules of the kidney. In the intestinal epithelium, they form striated border, while in the proximal convoluted tubules of the kidney, microvilli are seen forming the brush border appearance. Next, we go on to study the cilia, singularly called as a cilium. Cilia are cylindrical, motile structures on the surface of some epithelial cells. They are 5 to 10 micrometers long and 0.2 micrometer in diameter. They help in propulsive movement of fluid or particulate matter in one direction over the epithelial surface. And what we see here is a longitudinal section passing through a cilium. Where do we see cilia? They are seen in the trachea and bronchi, hair cells of vestibular apparatus in the inner ear, and along the uterine tube mucosa. Structure. Cilia have a cell membrane with a central pair of isolated microtubules surrounded by 9 pairs of microtubules. And the 2 microtubules of the peripheral pair are joined to each other. What we see here is a transverse section passing through a cilium. This is the central pair of microtubules, and these are the 9 doublets of microtubules which are present at the periphery, and outermost is the cell membrane. The cilia are inserted into basal bodies, which are small cylindrical structures at the apical pole just below the cell membrane. So what is seen here is structure of a cilium, and we see here the basal body which is seen just below the cell membrane of the apical pole of the epithelial cell. We now see differences between cilia and microvilli. Cilia are long processes measuring between 5 to 10 micrometer, while microvilli are short processes measuring 0.5 to 2 micrometer. Cilia are motile, while microvilli are non-motile. Cilia have a core of uniformly arranged microtubules in longitudinal orientation, 9 microtubule doublets located peripherally, and 2 single microtubules in the center. Microvilli have a core of thin microfilaments called actin. The actin filaments extend from the microvilli into the apical cytoplasm of the cell to form a terminal web, a complex network of actin filaments. Cilia help in propulsive movement of fluid or particulate matter in one direction over the epithelial surface. Microvilli greatly increase the surface area of absorption. Next, we want to study the stereocilia. Stereocilia are long, non-motile extensions of the cell membrane and should not be confused with true cilia. Length of the stereocilia is about 5 to 10 micrometer. They do not have microtubules in the core. And what we see here is a columnar cell showing stereocilia which are seen along the apical pole. Stereocilia increase the cell surface area, facilitates the movement of molecules into and out of the cell, and help in perception of various stimuli. And where do we see stereocilia in the body? They are seen in the epididymis and in the sensory hair cells of the cochlea. Next, we study the flagella. Flagellum means whip. Flagella have a whip-like appearance. It is a highly developed single cilium per cell. Length of the flagella is 15 to 30 micrometer. What we see here is structure of a mature spermatozoon having the head, neck, body and the tail. The tail of the spermatozoon is where we see flagella. So flagella differ from cilia by greater length of the flagella and 1 flagellum per cell. And as I said, where do we see flagella in the body? It is present in the human body only in the spermatozoa, and that is the tail of the spermatozoa, which is the flagellum. Thus, we have studied the initial part of the epithelium video, where we studied the basic introduction to epithelium, the derivation of the word epithelium, the definition of tissue, definition of epithelium, and then we went on to see the 4 specific surface modifications which are seen in the epithelial cells. In the next video, we will be seeing the classification of epithelium and we will be studying the simple epithelium. Thank you.